Thank you, Jim. It's such a pleasure to be here today to introduce today's special lecture with Dr. J. Craig Venter. Dr. Venter is regarded as one of the leading scientists of the 21st century for his numerous contributions to genomic research. I first met Craig when I was running a summer school in London. This was in the early 1990s at the beginning of the Human Genome Project. He came and gave a lecture to our students there. But interestingly, he had a special session with the students where he asked me and the other instructors not to come in the room, and he wanted to be with them and talk and get really good questions. And it was really great that he was able to do that today and met with some of our iGEM club students in my office, and they were able to discuss about their projects and interests. And since the early 1990s, I've been following Craig's great achievements. His innovation started with the development of express sequence tags, or ESTs, which were a revolutionary strategy for rapid gene discovery. He went on to implement a shotgun DNA sequencing approach that propelled the Human Genome Project towards completion. And more recently, he led the team that successfully created the first bacterial cell constructed entirely with a synthetic genome. His work to pursue the creation of new biological sources of food, fuel, vaccines, and clean water continue to make a positive impact on human health and the treatment of disease. Dr. Venter is a graduate of the University of California in San Diego, where he received both his bachelor's degree in biochemistry and a PhD in physiology and pharmacology. He has written more than 250 research articles and has numerous public honors and scientific awards, including the King Faisal International Award for Science in 2000, the Gardner Foundation International Award in 2002, and the United States National Medal of Science in 2008. Today, Dr. Venter serves as the chairman and CEO of J. Craig Venter Institute. This not-for-profit research organization is dedicated to human, microbial, plant, synthetic, and environmental genetic research and the exploration of social and ethical issues in genomics. He is also the founder and CEO of Synthetic Genomics, Inc., a company dedicated to commercializing genomic-driven solutions to address global needs. And just last week, Dr. Venter announced plans to start a new company, Human Longevity, that will focus on developing genomics-based approaches to slow aging and related disease. And when you get to my age, these types of companies become really interesting. Clearly, Craig Venter continues to blaze new trails in genomics. I am very much looking forward to his talk today, which is sure to be enlightening and thought-provoking. We will have the pleasure of learning from him as he presents Life at the Speed of Light, a discussion on how far and fast the science of genomics has come. Please join me in welcoming to Tufts University, Dr. J. Craig Venter.
Thank you very much, not only for the kind introduction, but for the invitation to join you here. It gets so boring with all that sunshine out on the West Coast. We've been watching winter on television, and occasionally it keeps things in perspective to come out and experience it for a few days. So thank you for that possibility. I want to start by introducing, and I don't know if the lights need to go down a little bit or not. This is our new institute building. This is at the Venter Institute on the University of California, San Diego campus. This is the world's first carbon neutral research building. So it's easy relatively to build a carbon neutral office building, but to do it for a research lab creates all kinds of new engineering challenges. And for example, the roof is a 0.5 megawatt power plant. There's major heat recycling. We use about 10% of the normal amount of water from recycling the occasional rainwater we get in San Diego. So it's created a totally new and wonderful environment to pursue this research. Now, talking about the life at the speed of light, there's two components to this. And when we started reading genomes, I call it digitizing biology, or converting the ACs, Gs, and Ts into the ones and zeros in the computer. This has been going on for some time. The first genome of any type was a DNA genome was read by Sanger and colleagues in 1977, the year before a group had sequenced an RNA phage. And this was a tremendous advance because it also came with a new technology for sequencing DNA. And this carried everybody for the next couple of decades. It was a long hiatus between 77 and 95 when my team sequenced the first genome of a living organism, that from Haemophilus influenzae, that used a shotgun method where we basically break the genome into millions of little pieces, sequence them, and reassemble them using unique computational algorithms. This was a big breakthrough, but it was only 5 years later that we were able to do this with a much larger genome, the haploid human genome that we reported in 2001, and then went on in 2007 to actually publish the first diploid genome. And this was a much greater challenge of separating the two haplotypes into a diploid genome, and a number of techniques that have gone into this, but this is a key part of the future of biology. As you heard, we just announced our new enterprise, Human Longevity, Inc., to scale up these processes from, cost $100 million and took 9 months to sequence the first half of a genome. And now with the exciting new technology that's come out of Illumina, based on work that originated here at Tufts, so you guys can claim ownership to the starting the second wave of genomics quite clearly, enables us to do potentially up to 100,000 genomes a year. So genomics is now not really the hard part of it. The hard part of it is trying to define what each and every one of you is, who you are, what you're made of, and can we standardize a phenotype, because we need digitized, standardized phenotypic information to align with the genome. And we're adding a number of unique components to this, and we have teams working on defining a human phenotype, groups including psychiatrists, as well as a range of physiologists to try and see if we can get to a standard definition. But the new X10 sequencing machine that we have a couple of now, they're starting to be delivered, will enable us to do up to 40,000 genomes a year. And we're building our facility to add on as many as 5 of these units. Changes the scale of this and puts all the pressure now on these other elements. Sequencing has changed a lot over the last 13 years, aside from technology making it fast and cheap. At the Venter Institute, we worked out how to sequence a genome on a single bacterial cell or a single human cell. We actually use this for haplotype phasing by isolating single sperm cells and sequencing their genomes. And each sperm is only a haploid genome, but no two sperm are alike, and no two eggs are alike. There's an average of 1 crossover per chromosome per sperm or egg, which is why there's basically an infinite number of combinations. But by just sequencing the genomes of 2 or 3 sperm, you get large segments of the haploid genome that it's pretty easy to then sort out into the 2 parental chromosomes. Everybody's asked the same questions at some stage in life. Did I get this trait from my mother, from my father, from my grandmother, my aunt? Did I give my nose to my child? Where did they get that nasty temper? It must be from my spouse. We all ask those questions, and now we can start to sort that out. And so by having the 2 parental chromosomes, we can start to ask these questions in a very comprehensive fashion. In fact, it becomes really important because in each gene that we get from each of our parents, we get different sets of variants. And so these are called compound heterozygotes. And these have not been measured or associated with anything in genetics to any serious extent. But it turns out you get about an order of magnitude more power and predictability by looking at these compound heterozygotes. And we did this even with my genome and sorting it out with disease-damaging mutations. So our goal is to try and change the nature of sequencing, the scale of sequencing, get down to haplotype phasing. And we're actually announced we're going to start this, trying to change the paradigm. So genome sequencing is the starting point for medicine. We're doing this with the Moores Cancer Center at UC San Diego, where every patient that comes into the Moores Cancer Center will get their genome done, and they'll get the full genome of their tumor. Obviously, this won't be a huge number of patients, on the order of 4,000 or 5,000, so it's a small portion of what we will be doing. We'll also be doing a number of, quote, normal people, from kids to super centenarians, trying to build up this comprehensive database, working towards 500,000 to 1 million. We're starting with cancer because genomics in cancer has changed a lot just in the last 3 years of getting driver mutations in a number of key genes that help determine therapy. For example, this ALK gene, mutations in that are probably the most important thing you could know about yourself if you have lung cancer, because roughly 4% of the population with lung cancer have these mutations. And if you have the mutations in ALK, Pfizer has a drug that has better than a 60% chance of eliminating your lung tumor. So knowledge becomes power. We only have about 24 of these actionable things in cancer. Our goal with these large databases is to change that to the thousands. So the new niche blockbusters are ones to treat a small number of patients, but charging an order of magnitude more for them. We don't know how long that's going to work for the drug companies, but as long as there's third-party payers and you get really incredible clinical results of solving things like lung cancer, it's probably going to keep going. Now, we're not alone in our bodies. We actually have more bacterial cells than we have human cells. This is called the microbiome, and this was discovered at our institute with doing shotgun sequencing of the gut, done by Karen Nelson, who's now the president of the institute. And this has become a very large field very quickly, and associations of changes in the microbiome are linked up to multiple diseases, including types of cancer. We have about 10 million microbial genes compared to our 20,000 something odd human genes. So obviously our genetic repertoire in our bacteria is much greater than it is with our own genes. And we can measure these in different parts of the body, and part of what we're setting up is high throughput diagnostics on the microbiome to be done in parallel with the human genome, because it turns out we have different genetic propensities for different microbes. Some of us have resistance for different microbes and viruses, such as the CCR5 gene providing resistance to HIV. At the same time, we're setting up a large proteomics unit to sequence proteins as markers for disease. And it turns out sequencing the proteins in urine turns out to be very effective in diagnosing and predicting the outcomes of a number of diseases. So trying to layer method after method, that each one exists in isolation. Proteome scientists talk to each other, metabolome scientists talk to each other, microbiome scientists talk to each other, genome scientists talk to each other. Physiologists don't talk to anybody. And so we're trying to put all these together because even understanding the metabolic potential, just putting the microbiome and the chemical repertoire together, it turns out we can make around 2,400 unique chemicals from our own physiology and biochemistry. So if we look in your bloodstream after a meal, what would we find? We'd find around 500 unique chemicals. Only about 60% of these are of human origin. 30% are from all the chemicals in the various species you consume in your diet. But about 10%, or maybe 50 chemicals circulating through your brain right now, are from bacterial metabolites of the food chemicals and your own human chemicals. Nobody's studied to see what effect they have on health, wellness, disease, daily outcomes, etc. So a metabolome in North Carolina can measure 2,400 chemicals using mass spec, and we'll be layering that data on as well. We've been trying to put some of these together, for example, in the field of iPS cells, because my genome was sequenced. A team in San Diego headed by Larry Goldstein made iPS stem cells and then differentiated those into neuronal stem cells and then to neurons. So there's a number of labs studying my neurons in culture in San Diego. But we sequence the genome at every step of this process, and there's about 1 million mutations in those neurons. And so I decided I didn't want them put back in my head without being able to alter them. So we're using genomics with our stem cell program, but we're trying to integrate all this data across in a new super database. These changes have started to be seen in stem cells. It doesn't matter whether they're fetal stem cells or iPS stem cells. And this is just tracing through what I just showed you of getting down to about 1 million changes in the cultured neurons. We're using some of our tools from synthetic genomics, some of the new CRISPR approaches, to go back and try and change en masse and rewrite the genetic code of stem cells so they can be used effectively for therapy. So more and more data, and it's going to scale up even more now with all this new human sequencing. Lots of ones and zeros in the computer that need to be sorted out. Part of the experiments that we started to do in 1995, when we sequenced the first 2 genomes, was ask some fundamental questions about the nature and origin of life, and got to the question, can we go the other way? Can we start with the ones and zeros in the computer and recapitulate life from those? We started just asking questions like how many genes are essential for life? What's the smallest number we could get to? Can we get to a minimal operating system of a cell that we could then build on in a systematic fashion? And then ultimately, to answer this, could we design and construct a genome chemically? And would the chemistry even allow us to do this? Nobody made larger than a 30 kb piece at the time, and there was only one of those that was made. That was a multi-year process. If we could make chromosomes, could we find a way to boot them up in a cell? So we started, decided to start small and start where genomics started with Phi-X. Clyde Hutchinson, who was part of the team, was actually in Sanger's lab and helped sequence the Phi-X genome and always kept a vial of Phi-X for some reason in his pocket all these years. So we could go back and resequence it and compare it to the original sequence. And then see if we could take the ones and zeros out of the computer and remake the Phi-X genome to see if we can get a virus out of it. Those of you who do DNA synthesis know it's an error-prone technique. It's n-1. The larger you make pieces, the more errors that are introduced. So Ham Smith, Clyde, and I spent a lot of time working out new ways for error correction. And in this 2-week process, we went from the digital code in the computer. We made small pieces, put them together, corrected them with error correction, and then inserted this piece of synthetic DNA into E. coli. And this was the actual first experiment where this happened. The E. coli immediately recognized the synthetic piece of DNA as normal DNA, read it, started producing the proteins. The viral proteins self-assembled to form the virus, and enough viral particles accumulated, killed the cells, and infected neighboring cells. So This is a lawn of E. coli where the virus has, in multiple settings, killed the E. coli cells. I call this a situation where the software is building its own hardware. People in the computer field would love it if all you had to do was write a new piece of software and a new computer popped out, but that is how biology works. And so we ended up with these infectious viral particles from a piece of chemically made software. Now, our goal wasn't to make a small virus, it was to make an entire chromosome, but we figured if we could make viral-sized pieces accurately, we could find a way to stitch them together to maybe make an entire chromosome. So we started down this route with Mycoplasma genitalium, which is a little under 600,000 base pairs. We designed 101 cassettes, and then something that looks like a Final Four playoff, we started putting these together. And this was very laborious. We had teams sitting there on the lab bench doing it. First, we had to make all the 6 kb pieces. And then we put 4 of those together to make 24 kb pieces. And each stage we had to grow them up in E. coli, sequence them, and make sure we're not introducing new errors, and then go on to the next stage. We put 3 of those together to get 72 kb pieces, and at this stage we were greatly in excess of the largest pieces ever made before. Again, we had to grow them in E. coli, sequence them, and make sure we hadn't introduced errors. And then went on to put 2 of those together to get a quarter genome molecules at 144,000 base pairs. But at this stage, E. coli didn't like these large synthetic chemical pieces of DNA, so we looked around for another system, and it turns out brewer's yeast is very good at taking up large pieces of DNA, and even better, as many of you know, it has a real nice homologous recombination system that if you have overlapping design pieces, it likes to put them together. So our solution was just putting basically the 4 synthetic quarter molecules together in yeast with a synthetic vector that just contains a yeast centromere. And yeast assembled these together for us very nicely. We isolated it and sequenced it, and that's what we reported in 2008. This was the first synthetic genome. And this was the largest molecule of a chemically defined structure ever made at that time. The chemistry continued to improve dramatically. Many of you have heard of Dan Gibson. Dan started as a postdoc in the lab and was taking part in these laborious assemblies and decided there had to be a better way. And so he found a way to take all the reactions and put them in a single test tube at a single temperature. And this was an amazing reaction because it worked even better than all the separate steps we were doing. And because it worked so well, we named it after him. It's the Gibson assembly. We're going to call it the Gibson cocktail, but somebody would want to put an onion in it. So in this, one of the advantages of this, other than its simplicity, allows it to be automated. So we can go from the digital code in the computer in a robotic fashion to make DNA. So this was progressing extremely well. The synthesis was getting faster, cheaper, and easier. And we focused our attention to our second team, which was trying to work out how to boot up a synthetic chromosome. Now with the virus where the DNA is infected with Phi-X, that's relatively simple, as you saw. With a chromosome, it's much more difficult. Bacterial chromosomes or any large chromosome are very brittle. You can't pipette an entire chromosome without fracturing it. So the team, headed by a French postdoc, Carol Latigue, and John Glass, shown here in the photos, developed ways where we'd isolate the chromosome in an agarose matrix. We could diffuse enzymes in there to chew up proteins, modify the DNA in this matrix, and then just dissolve the matrix when we're ready to put the genome into a cell. Because of the importance of this this is central to everything with synthetic genomics, I want to walk you through what we did with this converting one species into another. So we started with two mycoplasma species, Mycoplasma mycoides as a donor species and M. capricolum as the recipient cell. These are roughly as far apart as we are from mice, roughly 10% different in their genome, and also about 10% different in genome size. We isolated the chromosome from mycoides. When we first did this, we didn't know if there were going to be proteins necessary for chromosome transplantation, so we used harsh proteases to chew up all the proteins. We added cassettes to it so we could select for it with a TET system, and we added a LAC-Z system so it would turn the cells bright blue if this chromosome was activated. And we had to try different ways to get the chromosome out of the agarose blocks into the cell. But finally, we did this, and we have this sophisticated movie to show you what we think happened. We inserted the mycoides chromosome into the capricolum cell. And now we have something that's relatively rare in evolution, but it has happened before. We have the body of one species, and we have two sets of genetic software. What we think happened is, same with the Phi-X DNA, this new DNA started to be read immediately and produce proteins. Some of the early proteins produced were restriction enzymes that actually recognize the capricolum chromosome as foreign DNA and chewed it up. So now it's a little bit simpler. We have the body of one species and the genetic software of another. So what happened? In a very short period of time, we ended up with these bright blue cells. And when we interrogated them, there wasn't a single molecule left from the original species. Every protein in the cell derived from the new chromosome that we put in. All the other proteins were gone. All the antigens had totally changed over to be what was dictated by the new chromosome we put in the cell. So we call this, it was our first set of proof that life is a DNA software system. And if you change the software, you change the species. These are strange concepts to the way biology has been taught, but it explains an awful lot about life and evolution. So we were using the yeast as the final stage in our synthesis, but those of you who think about it will realize we had a unique problem. We had a bacterial chromosome in a eukaryotic cell, and we had to find ways to get it out of the eukaryotic cell to transplant it the same way we did with the chromosome that came right out of the bacteria. And to test this, we developed these methods for cloning entire bacterial chromosomes as yeast artificial chromosomes. And it was remarkably simple. It's a matter of adding the synthetic yeast centromere to a bacterial chromosome, and it converts it into a eukaryotic chromosome. So some steps in evolution could have been as simple as that, and these genomes are totally stable as an extra chromosome in yeast. So now we could try isolating the mycoides chromosome from the yeast cell. This is the native one, not a synthetic one yet, and trying to transplant it. The trouble is it didn't work. And this took us almost 2 years to resolve all the reasons this didn't work. But fundamentally, methylation was critical for protecting the genome. And when we isolated the chromosome from the bacterial cell, it had been methylated. That methylation protected it from the restriction enzymes in the capricolum cell. So what we did to prove this, we isolated the 6 methylases from the mycoides species. And if we methylated the DNA after we isolated it from yeast, all of a sudden we could do transplants. And to ultimately prove it, we removed the restriction enzymes from the capricolum genome. In that case, we could take native unmethylated DNA out of yeast and it would transplant. So these mismatches in methylation and restriction enzymes that you learned about in early biology were an essential step for either preventing or supporting major steps in evolution. When we first started scaling up to sequence a number of genomes, Several different ones were proposed, including the cholera genome. And the late Carl Woese argued there was no point in sequencing the cholera genome because it had the same 16S RNA basically as E. coli did. So it was just going to look like E. coli. I learned from my mentor, the late Nate Kaplan, the worst thing any scientist can do is talk themselves out of doing an experiment. So we did the experiment, and we actually found 2 chromosomes in cholera. One that, in fact, did look like E. coli, and the second chromosome looked nothing like E. coli. And it looks like it was acquired through a cell fusion or just taking up the DNA, and there were no restriction sites in that chromosome that were recognized by the enzymes in the cell. So it could have started out very much like E. coli, took up another chromosome and turned into a new species, cholera. We see lots of evidence for that in the organisms in the ocean, some with 4 or 5 chromosomes. And obviously the major steps in evolution were from taking entire bacterial species in, first as our mitochondria, but also in plants, taking them in, and that's how we got the chloroplast. So moving genomes around and moving cells around were key, very big steps in evolution versus it being just lots of little punctate mutations. So we wrote up this process on making the different changes of growing bacterial chromosomes in yeast, and this was a very effective circle because those of you who work on different strains of bacteria, most bacteria do not have genetic systems. Most people work on E. coli because they can, not because they want to. And now with this system, we can take a chromosome out of a bacteria without a genetic system and now use the yeast genetic system to make any kind of modifications in that genome, isolate it, methylate it if necessary, and transplant it to make a heavily modified cell. And you can just work around this circle. And the key thing is just adding this synthetic yeast centromere to the bacterial chromosome to allow it to function as a eukaryotic chromosome. So at this stage, we thought we'd solved everything. We had new synthesis paradigms with the Gibson assembly and our assembly in yeast. And we decided to take on synthesizing the 1.1 million base pair mycoides genome. And Dan Gibson led this team. We started off with 1 kb pieces. We put 10 of those together to make 10 kb pieces. We put 10 of those together to make 100 kb pieces. And we took the 11 100,000 base pair pieces, put them in yeast, that assembled the genome. We isolated it from yeast, transplanted it, and it didn't work. So again, it's like writing software. You have to have debugging software. So we had to create a biological equivalent of debugging software. In this case, we made segments of roughly 100 kb from the native genome where we could substitute each of our 100 kb synthetic pieces in to see if they all worked independently. 10 of the 11 worked, 1 would not support life. We had actually sequenced it with next-gen sequencing and went back and resequenced it with Sanger sequencing and found a single deletion in the DNA A gene that we then corrected, reassembled the entire genome, did the transplantation, and it worked. So 1 base pair being wrong out of 1.1 million, if it's in the right place, makes the difference between life and no life. So we were pretty excited when we finally got to the stage of this first cell. And one of the ways we set up to track, in fact, that these were only controlled by the synthetic chromosome is we set up watermarks in the genome. With the very first chromosome, we just used the single letter amino acid code to write things in the genome. It wasn't very creative, so we created a whole new code to allow us to write the entire English language with numbers and punctuation in the genetic code. And people have been doing this with the ASCII code, but not where biology is involved. So the code we developed puts in very frequent stop codons, because the last thing you'd want to do is put Tufts University into a genome and have that turn into a toxin that kills either the cell or somebody else. So our English language code puts in very frequent stop codons to not create new biology. So the first watermark contains the entire code to decode everything else. It also encoded a URL, being the first species to come out of a computer design. We thought it should have its own URL. If you translated this with the new code, you were instructed to send an email message to the species saying that you read its code. We had the names of 46 scientists that contributed to the work over the almost 15 years. And I came up with 3 quotations from the literature that I added to the genome. And I don't know if you can read those. I'll read them to you. To live, to err, to fall, to triumph, to recreate life out of life. That's from James Joyce. That seemed very appropriate. The second is from American Prometheus, Oppenheimer's biography, something he was told from when he was a young student from a teacher. See things not as they are, but as they might be. And the third was from Richard Feynman, what I cannot build, I cannot understand. After several people had translated this, and it became widely reported on the internet and the press, one of the first responses we got was a letter from James Joyce's attorney for his estate, asking us if we had copyright permission to use this quotation. James Joyce was dead. It was kind of hard to ask him. And also under US law, you can use up to a paragraph with attribution without having to get permission. So we dismissed that, and then we started getting emails from a Caltech scientist who said we'd misquoted Richard Feynman. And if you look on the internet, this is the main quotation you'll find. And so we asked him for evidence. He dug into the Caltech archives and sent us a photo of Feynman's blackboard with the original quotation, which is actually a much better quotation. What I cannot create, I do not understand. And somehow his biographer messed up the quotation, or he did himself on writing his book. So we've gone back to correct the genetic code. So the Feynman quotation will be correct, and Feynman can rest in peace as well. So what have we done since then? We've been working on trying to actually design a species in the computer for the first time. And in theory, this should be relatively simple. The problem is all the money that's spent understanding biology has not got us very far in understanding biology. So in every genome, there's a significant portion of genes of unknown function. At first, people tried to dismiss these and just say they were species-specific and had no importance, and then they started showing up being highly conserved across every species. And when we were doing gene knockouts, trying to understand the biology of these cells, we got down to about 100 genes of unknown function that all we knew is if we knocked out that gene, the cell died. So it kind of makes it hard to design things on first principles when you don't know what those first principles are. And I was talking to my uncle who just turned 90. He designed the Boeing 767, and I said, see, biology is very different from engineering because we don't know what the components do. And his response startled me. He goes, what makes you think we knew what things did? Apparently it was all trial by error in engineering as well. But so we're getting to the process. We're very close to a completely designed organism in the computer. But the other thing we've observed is evolution is pretty messy. Over close to 4 billion years, things get shuffled a lot, sort of like the hard drive on your computer if you don't defrag it. So we're developing a process for defragging genomes, trying to get them highly organized, sorting the genes by function, because if we're going to design things further, obviously it would be nice to have a cassette of whatever pathways you're putting in for energy metabolism, whether it's sugar metabolism, converting CO2 into methane, whatever we're using. It'd be nice to do those as cassette versus genes spread all over the genome. And this has not been a trivial process either, because in these circular genomes, there's multiple origins of replication, and things have to work in a symmetric fashion. So aside from just sorting the genes by function, we have to sort them by direction and orientation. So all those things that you learned in your biochemistry classes do actually play a significant role in biology. With these techniques becoming really standard for making large pieces of DNA, at SGI, they started a service called SGI DNA for making these really large constructs for a large number of partners. And one of the important things is, for somebody who started doing site-directed mutagenesis in a slow and cumbersome fashion, we can now design these combinatorial libraries to try and make new proteins. And we've done this successfully, making a new enzyme for a chemical pathway when we're trying to make the plastic used for making plastic bottles for sugar instead of from oil. The enzymes didn't exist. By just doing this combinatorial synthesis and change in design, we actually invented an enzyme that never existed before. And we're working on just cell design, trying to design new cells for new functions from scratch. We've been developing computer software for designing DNA software, and again, trying to get this in a modular fashion. When I was talking to your future iGEM team, I said next year we hope to start a contest of taking our minimal cell that's been defragged and making it available to schools around the country, and having a contest to see which school can add the best new evolutionary traits to this minimal cell. In theory, and it's been a theory that's been floating out there for centuries, you'll see it described in my book. If you started with basic protoplasm, you could recapitulate all of biology. In theory, we should be able to evolve this minimal cell into everything in the animal kingdom. We don't expect you to go too far too quickly, though. We're doing this ourselves, trying to change the biology of photosynthetic algae cells, which basically are a combination of a photosynthetic bacteria in a eukaryotic cell. So things are very compartmentalized. And so we're actually adding a whole new chromosome to the cytoplasm to do functions that would only exist, for example, in the chloroplast. We've been working on trying to modify photosynthetic efficiency. This is important in algae ponds where there's different levels of light. And we've managed to reduce the antenna size without any loss in efficiency. So these cells are about 3 times as efficient per chlorophyll molecule. Most of our effort in this space has been focused on new approaches in medicine, particularly with vaccines. This year, as a result of almost a 15-year collaboration between the Venter Institute and Novartis, we came up with Bexsero, which is a new, I think the world's first ever type B meningitis vaccine. And this recently got approved in Europe. This was the first genome-derived vaccine, because we started by sequencing the genome, then sorting out genes that weren't subject to phase variation, therefore would be stable across multiple species, put several of these antigens together to create this meningitis vaccine. And then it got tested very extensively in thousands of infants and kids in Europe, and that's why it was just approved, and the first doses in Europe were given last November. It's not approved in the US, but it's already been now given to large populations of students because of the outbreaks of type B meningitis, first at Princeton and then at UC Santa Barbara. So it's actually thrown the CDC and the FDA in kind of a quagmire of having the only effective vaccine being something that's been approved in Europe, and they've had trouble approving it here, but it's got approved twice now. And it's a very difficult situation because meningitis is one of those diseases that once you diagnose it, it's too late, but it's completely preventable. So if you can't get it here when you're on your summer vacations in Europe, I would urge you to get vaccinated until it's available here. And there's been this increasing series of meningitis B outbreaks. And in the irony of this could actually be selection from other effective vaccines, now making the meningitis B strain a much more widespread with new infections because there have been good A vaccines for some time now. Also with influenza, we've been part of the surveillance project using genome sequencing to track the emergence of new flu strains in pigs and birds and humans. And this has become very effective. And the reason for this concern is obviously the last major pandemic was the 1918 one, where about 3% of the world's population was killed from this, and that was before we had instant air travel anywhere in the world. So the worry with instant transportation, this would be much worse today. And so we focused on making new vaccines much faster. With the H1N1 pandemic that started in Mexico City, we were asked, I got a call from the mayor of Mexico City asking if we would sequence the isolate, but all these rules did not allow them to send the isolate out of Mexico to get sequenced. It was 9 months after the first outbreaks before there was a vaccine in the US. The pandemic had already peaked and was on its way down. So we tried to do it better this time. So the H7N9 outbreak that started in China, a group of Chinese scientists sequenced one of the isolates, posted it on the internet, and at the request of the US government and Novartis, we synthesized it. So it was first reported in March, and by the end of March and a week later, we had isolates that were available for the CDC and Novartis to start working on. And now there's been a synthetic vaccine made from our DNA constructs, and in a new facility. Most people don't realize the reason you get asked whether you're allergic to eggs when you get your vaccine. We use 300 million chicken eggs to produce the flu vaccine in this country, but now that's going to change because the new Novartis facility got approved using MDCK cells in the first mammalian cell culture, and this facility can produce 300 million doses. So they've now taken our H7N9 synthetic vaccine into clinical trials. It's done well on these, and the US government is now having Novartis stockpile this vaccine before we've had this first infection in the US. So for the first time, the country is actually ahead of the game. When H7N9 comes here, there'll be a vaccine immediately available to jump on it and prevent the spread. Let me close with how we're putting all this together. It's something I call biological teleportation. And it starts in a very simple fashion with a sending unit. And this was covered recently in a project we had with NASA. The LA Times and New York Times wrote a story on this. We're trying to solve the problem for sample return from Mars and other distant places. And the idea is relatively simple, is to sequence the genomes in situ and send the digital information back. So we tried this out in the Mojave Desert at the Mars test site. See, it looks just like Mars. And we have this really nice robot that wears blue gloves, and the robot picked up some samples. For example, if you're out in the Mojave Desert, you could turn over almost every rock and not find life. But if you see a piece of quartz, pick up the piece of quartz, and on the bottom side of it, you will find a whole series of algae growing. It's because just enough light filters through the quartz to give them the right wavelength of light. They're contained in a moisture environment away from the heat. And so that's where you can find a lot of diversity of life out in the desert, where most people would say there's nothing there. We took these into the Institute's laboratory bus equipped with an Illumina sequencer, sequenced this in a relatively short period of time, sent it all up to the cloud, and it turns out there were fungi, cyanobacteria, a whole range of organisms in this simple seeming micro environment. So that's the equivalent of our sending unit. You sequence in sight, you just send it up to the cloud. We've been working on the receiving unit, which we call a digital biological converter, that can just take a digital wave and convert it back into biology. We have, this was the prototype design, we've actually built it. This is funded by DARPA, and it's gone through all these extensive tests where all it gets sent is a digital wave, and it converts it in to DNA, RNA, and protein in a very relatively short period of time. So it's pretty exciting. A smaller version of it, just does part of this, will actually be on the market from Synthetic Genomics this year. I don't know if we're going to call it a GenBot, but this is a prototype of the instrument that can just automatically assemble up to 100 kp pieces in a complete automated version. But this takes us into the dual use category, and for the first time ever, we're designing instruments that have tremendous security built into them. We'll know from GPS where it is, we'll know from biometrics who's using it, and we'll know from having prepared the plates what will be assembled in them, and that if even the seal's broken on the plates, this can't be used to make something else. So we have to get it so the FBI and the government is comfortable with these being on the market, because each one of these is the equivalent of one of the DNA synthesis companies in terms of what its throughput will be. We hope this will, if your iGEM team gets one of these, nobody will be able to keep up with you guys. So it's very simple. If you can send things as a digital magnetic wave, the closest approach of Mars and Earth, we could get Martians back in as little as 4.3 minutes and reconstruct them in a spacesuit lab instead of having them splash down in the ocean. But most of you are probably not old enough to remember when the first astronauts returned from the moon. They were required to go into quarantine, but that's after they splashed down into the ocean, paraded around the deck of an aircraft carrier, and then were put into mobile homes for 2 weeks. So if they were really bringing the Andromeda strain with them, we would have all been in deep trouble. And one of the other rockets bringing samples back from sampling from asteroids also crashed into the desert. So we think our method of rebuilding the Martians and the aliens in a spacesuit lab will be much safer. So right now, you can download biology from your computer. We can make insulin and download it from the internet. We can make simple phage, and we can make a single self-replicating cells. So the challenge is, where does this go in the future, and what can be designed for the perfect app? Well, we think vaccines are going to be an important part of this. We've set up the first digital biological converter at the Novartis facility with funding from BARDA, that all we have to do is send a digital message to it, and it can make a new vaccine substrate that can go right into production in Novartis in a very short period of time. But the point is, if we can do that on one site, why not do it at multiple sites? And why not have smaller and smaller versions? And the ultimate way to head off a pandemic is stop them before they ever start. And so sometime in the future, you'll have a digital biological converter attached to your home computer, and you'll be able to download vaccines and certainly protein and RNA drugs from it. which creates a huge challenge for the regulatory environment. We asked for ethical review on our work before we ever started. Penn State took this, our University of Pennsylvania took this on and published this paper in 1999 in Science. The Sloan Foundation has been funding my institute along with MIT and a Washington think tank to look at security issues. And you can download these reports from jcvi.org. In 2010, when we announced the first synthetic cell, President Obama asked his new Bioethics Commission to take this on as their number one challenge. They wrote this report, which anybody in the iGEM field or anybody interested in this space should download this from the White House website and look at it. I think it's an excellent report dealing with the risk and benefit and the challenges for the scientific community in this new direction. And as anything that you missed, if you go to Amazon and buy my book, you can get it all. Thank you very much. So we're doing questions, and I guess you're starting.
Okay, so thank you for that brilliant overview. The question touches on the point that you ended at, which is, how do you think the general public will be enticed to entertain and to embrace some of these concepts that are ethically laden, ethically controversial? Some of the terminology might about when life begins, and gets the fundamental questions at the heart of society's conscience and technology's capabilities. So what do you think is the best way to reach, educate, and inform the public so they can become more literate about the power of these technologies that can be broadly applied and accepted and translated?
Well, for me, I find it's a good question. I thank you for that. The best way that I do it is I write books about it. I give public lectures about it. But it's challenging when you get outside the university environment. A recent poll showed that 60% of Americans don't know that tomatoes have DNA in them. And so somehow the whole education system has failed if that's where we are as a country. I can't personally solve that. Everybody here has to solve that. Everybody in the education system has to find a way to solve it. Or no amount of advanced scientific breakthroughs will be able to overcome a certain level of ignorance and resistance. So it is a huge challenge for our society, and it's a challenge when other governments are funding this space hundreds of times more than we are. And the U.S. either has to keep up its competitive edge and increase it, or we're going to end up way behind the rest of the world. So, and it's all linked together. Other questions?
My question, I guess, is related to that one. Most of us here are science students, and most of us plan on pursuing a career in the sciences. And for most of us, that's what we spend most of our time studying. But a lot of the issues you touch on and what we just talked about aren't hard science that we learn in the lab. So for us as students, what do you see as the 3 most important skills we should learn in our education and focus on in our careers other than science, to further science?
Well, it's tough because if you wander too much, you'll never graduate. But it's part of lifelong learning. You know, what I was told that basically what you get when you get your bachelor's degree is you've learned how to learn. That's probably all you've gotten out of it. Maybe some bad hangovers and other things, but you've learned how to learn, which you're supposed to apply constantly in the rest of your life. When you get an advanced degree, in theory, you learn how to do the rigors of science or the rigors of medicine. But again, applying on that lifelong learning. It's a challenge every day for everybody that has to be constantly renewed every day. And there's no one recipe for it other than paying attention and reading and seeing what the rest of the world really is doing. It's easy to disappear into your lab or your discipline, and that's necessary to get your degrees, right? Once you get your degrees, you got to broaden as quickly as you can, and that's why they want you to take some of these courses that really seem dumb to take, because they're not what you really want to learn about. It's getting exposure about the rest of these things. The difference between DIY biology and what you learn in an ethical lab is the right way to do it with the right ethics and the right safety concerns. So there is a value to doing this that you can't just get out of a cookbook. But other people looking in from the outside don't see it that way. So, not only do you have to educate yourself, everybody here has to constantly be an ambassador to the other 60% that don't know about DNA and that it doesn't exist in tomatoes. There's a question here, and I don't know where the mics are. And there's one up here.
So you mentioned that the software is all you need. So my question pertains to
move your microphone up. I can hear you, but
yeah. So my question pertains to the fact that the software is all you need. In some ways, if you think about it, there are additional components. For example, the membrane and other things which help you boot up the software.
yeah. If the software was all you needed, then DNA would be active on its own. Okay.
So I'm just considering the possibility, given that all the building blocks are available, can you make, for a lack of a better word, right-handed life instead of left-handed life, and just change the chirality of DNA that you're using?
Yeah, it's a good question, and scientists at NASA are very interested in that because they think it would be a shortcut to getting to a new genesis that would prove that life could start all kinds of different ways. I don't think it's needed, but certainly there's advantages for using the D enantiomers because they don't get degraded. So a number of peptide hormones people are using D amino acids for. Nobody's tried yet, and we have a pending proposal to see if we can make anti-life by making it with the other chirality. So we'll let you know. There's up in the back, yeah.
You mentioned computing and software a lot, like the defragmentation, for example, analogy. And I just wanted to hear your comments about the role that computing would have to do in kind of pushing your biological agenda, especially from an academic perspective as well.
Modern genomics would not be remotely possible without modern computing. If you even look at the problem we had of assembling the first bacterial genome, so we had 25,000 random sequences of roughly 500 base pairs in length. It would take a lot of monks sitting in a lot of monasteries a long time to piece all those together. With our first shotgun sequencing of the human genome, it was 25 million fragments. So, and that's just to get at one. To compare the 500,000 to million genomes we hope to have in databases within a decade, even existing computing is not adequate for that task. We're counting on computing and Moore's law to continue to drive things. They have to change in parallel. In fact, I had a fun debate with Gordon Moore about Moore's law, and I said DNA sequencing is changing much faster than Moore's law, so Venter's law trumps Moore's law. He said, but you're totally dependent on computing to do anything with your sequences, so Moore's law always wins. And unfortunately, he's right. The two go together. If we had 1999 computers today, there would have been no advance in genomics. We built the third largest computer in 1999. It was 1.5 teraflops and filled a room about this size. Now you can get a little card for your PC, a teraflop card. So things are changing, and we're counting on what we've even just announced with human longevity, that the computing will continue to change at the pace we need it to. There's a question here. Oh.
With the OSTP and the federal government requiring all of your federally funded data being open, I know that much of your data will go to perhaps NCBI, possibly PubMed Central. How is your lab dealing with making the rest of this data open? And where would you be considering repositing it?
So if we're talking about the patient data, we won't be opening that up to anybody because, in fact, there's other federal laws that require you to keep that private. In fact, we have to have that in highly secure databases that people will be able to log on and to use. It depends on the field. About 90% plus of the genes known to science have come from the deck of my sailboat, from the Sorcerer expedition. The Moore Foundation set up special databases to put this into because it was too much data for NCBI to handle. And so it really depends on the type of data and where it is. In my view, microarray data should never be made publicly available because it's not worth anything. It just clogs up databases. So I think this old notion that derived out of the early days of gene sequencing and protein sequencing, where everything had to be deposited in databases, simply cannot keep up with science or even the ethical requirements with patient samples.
You mentioned earlier about the funding problem in the United States, or that our government doesn't seem as committed as other governments to increasing science capability in this country, as this is a room, as Maddie referenced, full of people who are interested in being future scientists. Where do we go in this atmosphere where it looks like you could be a postdoc forever, or you could be a grad student forever, and not like, but Sanger published 3 articles in 20 years, 3 of the most seminal articles that make this room possible, but in this academic environment, it seems like someone would have said you only published 3 articles in 20 years.
Yeah. I published 21 articles in my 3 years as a graduate student, so I did the opposite. But and I didn't do a postdoc. So but the rules are changing, and it gets more challenging. And it's not that we don't have sufficient funds in our federal science budget. We have massive amounts of money in the federal science budget. We just don't spend it very well. We sort of let the inmates run the prison. NSF calls everybody that gets money from them their clients, and they let the clients make the decisions about who gets the money. I was on a Senate hearing in the early days of the Genome Project with Jim Watson, and he said to the senators, "I know how to cut the science budget by 90%. And it was, I think, Al Gore who said to him, "Please, Dr. Watson, tell us how to accomplish that." And Watson said, "You just give it to the scientists, they're going to make the breakthroughs." And the problem is we don't have a method that selects how to do that, but we have a method that deselects from it because we encourage people not to make breakthroughs in science. So it's not that there's not sufficient funds in the country. We got to learn how to spend it a whole lot better. The scientific community, the public should be outraged there's not 100 times as many breakthroughs for the amount of money we're spending. That's a lot of Me Too, very limited advances with science. Any of you know, if you what's the main criticism you get back that kills your grant is you're too ambitious. I don't see how people can be too ambitious in science. So you have to help change the rules. In my case, I just went around them and created my own funding sources by inventing ideas where people would give me money to do what I wanted to do. it seems to still be working at age 67. So we'll see if it keeps working. Or you can do what he did, make a great invention, and people just throw money at you for the rest of your life, right?
Hi.
Yes.
My question is, you equated the computer language to, in some aspects, to the biological language. And I guess I was wondering what your thoughts were in terms of, could there possibly be more than one language in biology? Particularly because if you look at the complexity of animals, multicellular animals, the complexity of the genome in terms of number of genes doesn't seem to be proportional to that. So I was just wondering what your comment would be on that.
What department are you in?
Biology.
Really? Okay. It still surprises me. The biggest surprise to the scientific community and the rest of the world when we published the first human genome was there was only 20,000 some odd genes, because the genetics community and just simplistic views of thinking had people wanting to have one gene for every human trait. And if that's your thinking, then there has to be a whole lot of genes. And some people get very offended, that we only have 6 billion letters in our genome and the lily plant has 91 billion. Right? I'm not particularly offended by that, but I haven't lost an argument to a lily yet. I don't know how to explain combinatorial biology to somebody who thinks in a linear fashion, right? I mean, it's you can't compare the two. You have 100 trillion cells, so you have 100 trillion different combinations of your 20,000 some odd genes. You work out how many combinations that is. That's a lot of combinations. Having twice as many genes won't make you twice as gorgeous, okay? So it's a combinatorial issue.
So this, that was loud. So this question kind of relates back to
A
funding.
little bit closer to your speaking
So this
part.
last question goes back to funding and these large projects that oftentimes when we apply for grants, reviewers say are just too large. And you've talked about all these great projects and they're huge, and you want to sequence 40,000 genomes a year. If you brought that up before, people would say that's just impossible. Don't even try to do it. And now, because you have the funding, you can do that. As a grad student, we're kind of, I don't want to say pigeonholed, but we're told to think smaller and not try and take on these huge projects or even think about these massive projects, because there's just no way we can do it, or oftentimes a lab or a whole department can't do it. So how do we transition to having these grandiose ideas and ideas when we're trained to really just think small? And that's just not a criticism of our department or Tufts University as a whole. It's a
Well, if that's flattery, please don't flatter me that same way. Look, there is a rationale for it. It is when you're learning this process of how to do science, part of the rigor for doing it the right way is to pick something and do it in an absurdly thorough fashion, right? That's part of learning the art of doing it. The hope is you'll never do that again in your life, right? And that you'll take that art, that skill set, that set of rules, that set of ethics, and you apply it on a larger scale. I couldn't get the government to give me money to do any of these genomes. Ham Smith and I, even though he had a Nobel Prize from 1978, we wrote up this notion we had for doing shotgun sequencing of a bacterial genome, and it was turned down with extreme prejudice. And even when we had it 90% done, as I cover in my first book, I called Francis Collins and said, look, this is going to be a big embarrassment to you guys. Just fund it, and you can help take credit for it. They were so sure it wouldn't work, they wouldn't fund it. They certainly wouldn't fund us to do the human genome. So, we had to go out and find independent funding for it. And I've been lucky in that regard. At times it's better to be lucky than smart, but you can create your own luck. And if you can't get what you want from one source, find and take part in a big project and learn how to do it. Good science is like making good movies. If you get the chance to make one movie and it's a blockbuster, and you want money to make another one, it's pretty good chances you'll get it. And if you get a track record of making 5 or 6 blockbuster movies, you can actually have a failure and you'll still get money to make your next one. If you never make a blockbuster or you try and they're all failures, you're doomed. You're not going to get that money again. So, if you can find a way to break out of it, do it and make sure it works. And that's what we've done at every stage. And, now it's less difficult to ask people for $70 million for a big idea because I've made other big ideas work. If I tried that right out of graduate school, I don't think I would have gotten very far. So it's a matter of building the skill set. It's a matter of building the scientific credentials that you have the discipline, you have the skill set, you have the team building things. That's one thing that's not taught in graduate school. That's absolutely essential for success in science. It's how to build and run and manage a successful team. In fact, academia tells you you don't even want to be on a team. You got to be your own little academic lab. That's going away, Mr. President. The future, at the Venter Institute, nobody has their own lab. They don't have their own equipment. We have a Nobel Prize winner that has about this much bench, and he can be working next to a high school student. But we try to get them all the resources they need to do their science. And that's contrary to the single investigator method of most of our universities of you have to have your own space, you have to have your own equipment. There's this tremendous redundancy in science. That's where a lot of the money goes in science that's wasted. So all these things sort of fit together, but you look like a bright guy. I'm sure you can come up with an idea and convince somebody to fund your first experiment after you get your PhD from doing the really boring stuff.
Last question.
Last question, uh-oh.
Oh dear. I'm not in your field, so forgive me if I have a sort of simplistic view of what you're doing. And it appeared to me that you were mainly taking living things and transforming them into other sorts of living things. And I wondered if you'd been able to create a living thing totally out of non-living substances, like say a cell. And if so, how would you reflect on its religious implications?
Yeah. So that was, to sum it up, that's sort of one of the criticisms early on with our 2010 announcement of, I pride myself of being the only scientist in modern times that got comments from the president and the Pope in the same day. The president congratulated us. The Pope said we only changed one of life's motors. It's not my job or desire to alter people's religious beliefs. So to me, that's completely irrelevant to science. People like Jack Sozak across town at Harvard is trying to create life from scratch. Lipid vesicles, RNA molecules. That's not what we're doing. We're building on top of 4 billion years of evolution, which gotten us to a very advanced stage, showing that we're a software-driven species, and you can put in totally new software and change any species into some other species. We could take a cell from your arm, change the genome, and convert it into a pig cell. Doesn't mean we can convert you into a pig, but biology is software-driven, and it's an important aspect to realize that it's software-driven. How we got to this advanced stage of evolution over 4 billion years, maybe it came from something like Jack Sozak's RNA molecules and lipid vesicles, it's just started separating and getting more and more complex over time. It's a very different question. But it's one that I address in my book, and we try to answer somewhat ourselves, because we were trying to put proteins together to see if we could, quote, make a cell from scratch. Because the theory of cell biology is all cellular life derives from other cells. But it's like trying to say you built a new car by buying a Chevy Corvette engine, buying a chassis from someplace else, buying a fiberglass shell from someplace else, putting it together and say you made a new car. The ribosome is the most complex part of our cells. It's this huge complex that makes our proteins. We can take a ribosome complex and we can put it in a lipid vesicle and get it to work, but that's not creating life from scratch. So the closest one I know to trying to do that is what Jack's doing in his lab at Harvard. I think it's more important to understand that all the information about you is contained in your software, and changes in that software will change who you are and what your life outcomes are. Yeah. Thank you for the question.